Good afternoon. Today we are going to discuss about action potential. So we have already discussed about resting membrane potential. The potential difference across a cell at rest, if recorded by different two electrodes, one placed inside the cell and other outside the cell, gives the resting membrane potential. We have seen that the resting membrane potential for different tissue is different. For a neuron, it is minus 70 millivolt. For skeletal and cardiac muscle, it is minus 90 millivolt. For smooth muscle, it can vary between minus 25 to minus 65 millivolt. In the pacemaker tissues of the cardiac muscle, it can be between minus 55 to minus 65 millivolt. So it varies from one tissue to another. We have also seen about the factors responsible for genesis of resting membrane potential. Let's just revise the first is the difference in concentration and permeability of different ions across the cell. Potassium is much more permeable to the cell at rest as compared to sodium ions. Then comes the equilibrium potential and the Nernst potential and the Nernst equation, where we consider that that membrane is permeable to only ion. So the ionic and the electrical concentration gradients are two forces acting opposite to one another. Potassium is more inside the cell. It is less outside the cell. So potassium tries to move out of the cell. Inside the cell is negative. This prevents the potassium to move out of the cell. So these are two forces, electrical and concentration gradient acting against one another. A time will come when these forces will equalize one another and there will be no net diffusion of ions. This is called the Nernst potential or the equilibrium potential for that ion. But in this instance, we only consider that the membrane is permeable to only one ion. Then comes the Goldman-Hodgkin-Cadge equation or the Goldman constant field equation, where we consider all ions at a time. And here also we consider the permeability and concentration of different ions. Then comes the Donnan equilibrium and the Donnan equation. There is protein present inside the cell and the cell membrane is impermeable to the negatively charged protein. So the permeable ions will behave in such a way to maintain equilibrium, which can be predicted by this equation. Then comes the sodium-potassium ATPase pump, which contributes minus 4 milli equivalent to the resting membrane potential. Though it contributes minus 4 equivalent to the resting membrane potential, without the action of the pump, this electrogenic pump, what will happen? Sodium will not move out of the cell and potassium will not move into the cell. So if this pump stops, number of ions inside the cell will increase. As a result, there will be osmosis and water will move into the cell, which is called the cell to burst. So it is also helpful for regulation of cell volume. Then we have learned that with the cathode ray oscilloscope, we can measure the resting membrane potential as well as action potential. Here you can see one electrode is placed inside and the other outside the cell, and these are connected with electronic amplifier Then there is an electronic stimulator which stimulates the nerve or the excitable tissue. Then there is electronic sweep circuit which is connected with horizontal plate, and the electronic amplifier is connected with vertical plates. And with electrical gun, there is electron beams coming, and these are deflected by the horizontal and the vertical plates, and they can be recorded on the screen. Initial part out here, you can see there is a stimulus artifact. It is due to leakage of current, and when we apply a threshold stimulus, finally an action potential is generated. The action potential may be defined as the brief sequence of changes which occur in the resting membrane potential when stimulated by a threshold stimulus. So action potential may be defined as the brief sequences of changes which occur in the resting membrane potential when stimulated by a threshold stimulus. Now let's come to this picture. Subthreshold stimuli do not produce an action potential, but they have got an effect on the membrane potential. We can demonstrate this by placing recording electrodes within a few millimeters of a stimulating electrode and applying subthreshold stimuli of fixed duration. Application of such currents lead to a localized depolarizing potential change that rises sharply and decays exponentially with time. The magnitude of this response drops off rapidly as the distance between the stimulating and recording electrodes is increased. If an anodal current produces a hyperpolarizing potential change of similar duration. So there can be depolarizing currents and hyperpolarizing current, which can produce a change in the membrane potential. The potential changes are called electrotonic potential. So you can see here the resting membrane potential was minus 70 millivolt in the neuron. For the first 7 millivolt is the electrotonic potential, electrotonic conduction out here occurs. Then the next 7 millivolt is the local response. Here the membrane starts participating, that is channels open up. At minus 55 millivolt, you can see in this picture is the firing level, and there is generation of an action potential. It is also possible to determine the minimal intensity of stimulating current, that is threshold intensity, that acting for a given duration will just produce an action potential. The threshold intensity varies with the duration. With weak stimuli, it is long, and with strong stimuli, it is short. The relation between strength and duration of threshold stimulus is called the strength duration curve. Slowly rising currents fail to fire the nerve because the nerve adapts to the applied stimulus, a process called adaptation. We have seen if we apply a slowly rising current, then what happens? The sodium channels and potassium channels both open. The sodium channels are fast to open and fast to close. The potassium channels are slow to open, but they remain open for longer interval. So the process of adaptation occurs in this case. Once threshold intensity is reached, a full-fledged action potential is produced. If you further increase the intensity of stimulus, no increment or no other change in action potential occurs, as long as the experimental condition remains constant. The action potential fails to occur if the stimulus is threshold in its magnitude, and it occurs with constant amplitude and form regardless of the strength of the stimulus if the stimulus is at or above the threshold intensity. So the action potential is all or none in character. So the character of the action potential is all or none in character. Now let us discuss about the changes in excitability during electrotonic potentials and the action potential. During the action potential as well as during electrotonic potential and the local response, the threshold of neuron to stimulation changes. Hyperpolarizing responses elevate the threshold while the depolarizing potential lower it, and as they move the membrane closer to the firing level. During the local response, the threshold is lowered, but during the rising and much of the falling phases of the spike potential, the neuron is refractory to stimulation. The refractory period is divided into absolute refractory period, corresponding to the period from the time of the firing level is reached until repolarization is about 1/3 complete. A relative refractory period then lasts from this point to start off after depolarization. So you can see that so this is the picture of an action potential. So first is the firing level is reached, then there is generation of an action potential, and finally there is repolarization of the membrane. So from the firing level till the 2/3 of the repolarization, what happens? This is the absolute refractory period. So what happens? Until the repolarization is reached 1/3 complete, it is absolute refractory period. A relative refractory period then lasts from this point to the start of after hyperpolarization. During the absolute refractory period, no matter however strong stimulus you apply, you will not be able to generate an action potential. But in the relative refractory period, if you increase the strength of the stimulus, you will be able to generate an action potential. Here in this picture, you can see that the sodium conductance of the membrane is short-lived and the potassium conductance is long-lived. See in this picture, there are opening and closing of different channels. For production of an action potential, you require a critical number of sodium channels to open. The sodium channel, as we have discussed, has got 3 stages. One is the closed stage, followed by the inactivation state, followed by the open state, until and unless the sodium channels are closed. However, the strength of stimulus you increase, you will not be able to generate an action potential. So this period is the absolute refractory period. Absolute refractory period. So this is up to about 1/3 of repolarization until complete. This is the absolute refractory period. Now let's see the different steps out in this picture. So just follow the picture. We will discuss the different steps. See, initially there is the resting membrane potential. Now you have applied a threshold current. So what is happening? Now the membrane potential is rising. You can see follow the step 2. So what is happening? The membrane potential is rising. The voltage gated sodium channels are opening. We told that the sodium channels are fast to open and fast to close, while the potassium channels are slow to open, but they remain open for longer interval. So the activity of sodium channels here is more. And once sodium channels open, they follow a positive feedback mechanism, which is called the Hodgkin cycle, while the potassium channels, they follow a negative feedback mechanism. Now you can see that the membrane potential is rising. It has reached the firing level and the membrane has fired. But the membrane doesn't reach the equilibrium potential for sodium, because sodium channel opening is voltage and time dependent. So after a certain voltage, they will get inactivated. And when they get inactivated, however, the strength of the current you increase, as we have discussed, you will not be able to open this channel until they are closed. If you increase the strength of the current, you will not be able to open the channel. So you can see above the 0, there is an overshoot. After phase 3, you can see there is phase 4. The blue color, the green color one is the action potential. The red color is the sodium conductance of the membrane, and the violet color is the potassium conductance of the membrane in this picture. Then you can see there is potassium channels are open. So what is happening? The membrane or conductance for potassium is there. As a result, the membrane is repolarizing because the voltage gated potassium channels open. These factors contribute to the repolarization. The opening of voltage gated potassium channel is slower and more prolonged than the sodium channels. Much of the potassium conductance comes after the increase in sodium conductance. That is your step 5. You can see the picture, step 5. The net movement of positive charge out of the cell due to potassium efflux at this time helps complete the process of repolarization. The slow return of potassium channel to the closed state also explains the after hyperpolarization. You can see step 6, followed by return to the resting membrane potential. That is step 7. So step 1, 2, 3, 4, 5, 6, and 7. These are the different steps of the action potential. Decreasing the external sodium concentration reduces the size of action potential, but has little effect on the resting membrane potential. In contrast, since the resting membrane potential is close to the equilibrium for potassium, changes in the external concentration of potassium ions can have major effects on the resting membrane potential. If extracellular potassium increases, that is hyperkalemia, the resting membrane potential moves closer to the threshold for eliciting an action potential. Thus, the neuron becomes more excitable. If the extracellular level of potassium is decreased, that is hypokalemia, the membrane potential is reduced and the neuron is hyperpolarized. We have already discussed that the sodium entry, it follows a positive feedback mechanism, while the potassium channels, they follow a negative feedback mechanisms. Calcium can affect the membrane potential through both channel movement and membrane interaction. A decrease in extracellular calcium concentration increases the excitability of the nerve and muscle cells by decreasing the amount of depolarization necessary to initiate the changes in the sodium and potassium conductance that produce the action potential. An increase in extracellular calcium concentration can stabilize the membrane by decreasing the excitability. So, a decrease in extracellular calcium concentration increases the excitability of the membrane, while an increase in extracellular calcium concentration can stabilize the membrane by decreasing the excitability of the membrane. You see in this picture, minus 70 millivolt is the resting membrane potential. Then you have applied a stimulus which were not threshold. So subthreshold stimulus, there was a change in the membrane voltage, but this was not enough to generate an action potential. Then you applied a threshold current. So firing level was reached at minus 55. Then due to opening of sodium channels, Hodgkin cycle, there was depolarization of membrane. There was some overshoot, but it didn't reach the equilibrium potential of sodium because the sodium channels are fast to open and fast to close, and they are voltage and time dependent. There are three stages as we have discussed. One is the open state, followed by inactivation state and closed state. If the channels become, once they become inactive, however the strength of current you increase, you cannot generate an action potential. The sodium channels are fast to open and fast to close, while the potassium channels are slow to open but remain open for longer intervals. So sodium channels have become inactivated, and now in the overshoot, after the overshoot, and the potassium channels are open, so the membrane repolarizes. The potassium conductance of the membrane remains for longer intervals, so there is some hyperpolarization of membrane, followed by returning to the resting membrane potential. So action potentials are the primary electrical responses of neurons and other excitable tissues, and they are the main form for communication within the nervous system. They are mainly due to changes in conductance of ions across the cell membrane. The electrical events in neurons are rapid, being measured in milliseconds, and the potential changes are small and measured in millivolts. You can see here, same picture, action potential, overshoot, depolarization, repolarization, then hyperpolarizing some part, finally returning back to the resting state. You can record all this with the cathode ray oscilloscope. So initially there is some leakage of current that is responsible for the stimulus active artifact. Then you can see there is a firing level reached, followed by depolarization. From 0, there is overshoot, but doesn't reach the equilibrium potential for sodium because sodium channels are voltage and time dependent. And for generation of action potential, you require a critical number of sodium channels to open. Then you see there is repolarization of the membrane, hyperpolarization, followed by bringing back to the resting phase. This phase, this after depolarization, what happens when potassium is moving out of the cell? So the concentration gradient is gradually dissipating. So in this part of after depolarization, the slope decreases and the efflux of potassium also decreases. This is responsible for this phase. We have already discussed about electrotonic potential, local response, and the firing level. At firing level, critical number of sodium channels open due to application of threshold stimulus. Electrotonic potentials and local responses, they are non-propagated, and here threshold stimulus is not applied. So you can see here local response, resting membrane potential, firing level, propagated action potential. The local response is a graded potential. It can be either depolarizing or hyperpolarizing, and it can be summated. Action potential is always a depolarizing potential, and it follows all or none law. That is, when a threshold stimulus is applied, the membrane will respond with its ultimate vigor, and if you further increase the strength of the stimulus, the response will not be greater. So it is of all or none character. Changes in excitability during electrotonic potential and the action potential. During the action potential as well as during electrotonic potential and the local response, the threshold of neurons to stimuli changes. So hyperpolarizing responses, as we have discussed, will elevate the threshold, while depolarizing potential lower it, and they move the membrane potential closer to the firing level. You can see here again the same picture. I am repeating them repeatedly because for you to remember the sodium conductance of membrane, the potassium conductance of membrane, and the action potential, their comparison. You can see the absolute refractory period, the relative refractory period, and the effective refractory period. So there is overshoot, sodium conductance, potassium conductance of the membrane. Activation and inactivation of sodium and potassium channels, and potassium conductance and sodium conductance of the membrane you can follow in this picture. You can see the potential changes, local response, the spike potential, the after depolarization, the hyperpolarization, the supernormal stage, the refractory period, the subnormal period. So action potential, you can tell it occurs in an excitable tissue, example, neuron, muscle fibers. It is a threshold current needs to be given, and the sodium channels, they follow a positive feedback mechanism, that is the Hodgkin's cycle. The action potential, it is an all or none response and lasts for a few milliseconds. There are states of depolarization and repolarization. The majority of all action potentials are generated in the axon hedon because, or the initial segment of the axon, because the number of channels are more 350 to 500 in the initial segment. So voltage gated sodium channels are highly concentrated out there. This triggers the action potential. An action potential is generated due to membrane potential reaching threshold. This minus 55 is the firing level. Steps involved are membrane depolarization due to opening of sodium channels, then sodium channel inactivation, potassium channels now are active. So membrane finally repolarizes and return of normal permeability fiber. If depolarization at a spot on the cell reaches the threshold voltage, the reduced voltage, which opens up hundreds of voltage gated sodium channel in the portion of the plasma membrane. During the millisecond that the channels remain open, lots of sodium rush into the cell, and this follows a positive feedback mechanism. The action potential or self-propagating potential, that means that one action potential automatically triggers the neighboring membrane areas to produce an action potential. Thus, once threshold is reached, action potential always propagate down the axon to the synaptic end or the secretory regions of the synapse. In this picture, you can see the feedback control in voltage gated ion channels in the membrane. Sodium channels exert a positive feedback mechanism. So you can see there is a depolarizing stimulus, there is opening of voltage gated sodium channel. So this follows a positive feedback mechanism, increasing conductance of sodium, which causes more and more sodium channel to open and more depolarization of membrane. It stops until the sodium channels get inactivated. This is the Hodgkin's cycle. Potassium channels, they exert negative feedback. Starting, there is depolarization of membrane by sodium influx, opening of voltage gated potassium channel, increased potassium conductance, increase of potassium flow out of the cell, repolarization of membrane. This follows a negative feedback mechanism. The actual process of the action potential occurs in 4-step consecutive, but some are overlapping. There is opening and closing of different channels during this phase. So the action potential is usually generated in the initial segment of the axon because of the distribution of channel is very high here. That is 350 to 500 in the initial segment of the axon. The number of sodium channel per square micrometer of membrane in myelinated mammalian neurons. Now we come to orthodromic and antidromic conduction. The axon can conduct in either direction. When an action potential is initiated in the middle of the axon, two impulses traveling in opposite directions are set up by electrotronic depolarization on either side of the initial current sink. In the natural situation, impulses pass in one direction only, that is, from synaptic junctions along the receptors, and receptors finally to the terminals, finally to the postsynaptic membrane, the receptors. Such conduction is called orthodromic conduction. Conduction in the opposite direction is called antidromic conduction. So you can see in this picture stimulus. One is the orthodromic conduction, another is the antidromic conduction. Here, what happens in the antidromic, two depolarization cancel one another, but in the orthodromic conduction, progressive depolarization occurs to CNS for perception. So again, you can see the stimulus, antidromic conduction, orthodromic conduction. The two depolarizing potential, they cancel one another because there is somatic motor input from the spinal cord or brain. Here you can see progressive depolarization to target tissue and receptor that occur. So motor output to muscle or gland that occurs. Generation of action potential in the initial segment from the resting membrane potential, threshold, then firing level, that is a firing level, then there is overshoot, followed by repolarization of the membrane. Some hyperpolarization because potassium conductance still remains, finally bringing back to the resting membrane potential. All these you can record with the cathode ray oscilloscope. Again, in this picture, you can see depolarization of membrane, overshoot, first reaching up at the firing level -55, followed by overshoot, the depolarization, sodium channels become inactive, repolarization due to the opening of potassium channels, inactivation of sodium channels. Finally, there is some hyperpolarization, bringing back to the finally to the resting membrane potential. Now we should also learn about chronaxy, Riobes, and utilization time. Riobes is the minimum strength of current required to stimulate the tissue. Utilization time is the time required by a Riobesic current. That is the time taken by the Riobesic current to stimulate the tissue. And when you increase the strength of the current to twice that of the Riobes, the time taken by the tissue to response is your chronaxy. So you can graphically represent them with a strength duration curve. You can see here number of motor units recruitment, intensity of current, frequency, and placement of electrode. SD curve is graphical representation of these things. Here you can see the voltage, and here you can see the duration of the current. So we have already told that it is possible to determine the minimal intensity of stimulating current, that is the threshold intensity, that acting for a given duration will just produce an action potential. The threshold intensity varies with the duration. With weak current, it is long. With strong stimulus, it is short. The relationship between strength and duration of threshold stimulus can be represented by the strength duration curve. Again, you can see here, this is the Riobesic current. The time taken is the utilization time, and chronaxy is the time taken by twice the Riobesic current for the tissue to response. So utilization time, chronaxy, and Leo base. So again, we are revising the action potential in the neuron, minus 70 resting millivolt, sodium channel opening, minus 55 firing level, critical number sodium channels have opened, sodium channel opening and inactivation and closing all are voltage and time dependent. There is some overshoot. Sodium channels have become inactivated. Potassium channels are open. Membrane repolarizes. There is some hyperpolarization because potassium conductance remains. And finally, there is bringing back to the resting membrane potential. Fine potassium channel opening and potassium channels, they follow a negative feedback mechanism cycle, and the sodium channel, they follow a positive feedback mechanism. The first state is the resting state, where all active ion channels are closed. Almost all voltage gated sodium channels and potassium channels are closed. Only the leaky channels are open. And you know, the potassium conductance of membrane is much more as compared to the sodium conductance at rest. So more is of the resting membrane potential is contributed by potassium flow. Potassium conductance of membrane is much more through leaky channels at rest. The depolarization of the membrane is caused by in this instant in the neuron, by the opening of sodium channels, and sodium channels, they follow positive feedback mechanism. The repolarization is brought by the opening of potassium channels. You can see this picture, opening state of sodium channel, inactivation state. You can see opening state, inactivation state of sodium channel, and the closed state of sodium channels. So we will discuss rest of the action potential in the next day. Go back and study this. Thank you. Bye.